very much, Liz, for the introduction and for the invitation as well to speak. It's a great pleasure to be here. And yeah, my talk today is using the odour of bacteria to tackle acute oak decline. So it's obviously quite an obscure title, but hopefully over the next 20-25 minutes or so, I'm going to provide a bit of clarity on what I mean exactly by that. So first of all, I want to just sort of state the importance of oak trees. So oak trees support a wealth of biodiversity and provide a habitat for a range of insect and animal species. They're sort of key components of the landscape and also crucial components of the timber industry. So it's quite crucial that we maintain their health. But as with all sort of plants, they're sort of subjected to a range of biotic and abiotic stresses, which can lead to the onset of diseases. One specific disease, which will come as no surprise to you, is acute oak decline. And acute oak decline is characterised by a range of symptoms. Some of the main symptoms, which you can see here from this middle picture, is the presence of these necrotic lesions, which kind of look like somebody's got a paintbrush and kind of gone down the tree with it. Some other sort of symptoms include this sort of inner bark necrosis and cracking of the bark as well. And this combination of symptoms can lead to the rapid decline of oak trees within about 3 to 5 years having sort of obtained the disease. And some previous work from collaborators at Forest Research have looked at what's causing these bleeds. So here we've got a zoomed in picture of the bleeds. And what they've shown is that there's 3 bacterial species which are commonly associated with these bleeds. And those 3 bacteria are Bruneria goodwinii, Rhinella victoriana, and Gibberella querceneckens. And yeah, these sort of 3 bacteria kind of work together to cause these sort of bleeds and the onset of the disease. And what they've also shown is actually if you take these bacteria in isolation and put them onto a healthy tree, they will actually form these lesions as well. So we're pretty confident then that these are the cause of this disease. And in terms of the sort of distribution of acute oak decline in the UK, this is a figure from Forest Research, who do a lot of work in the monitoring of acute oak decline. We can see that where a purple sort of circle indicates the presence of acute oak decline, quite a lot of localization around the southeast of England. But what we're starting to see over the years, so this data is from 2006 to 2023, is a sort of northward shift in the presence of acute oak decline, with recent reports having detected it in places like Liverpool and Manchester. And in terms of across the country, it's also now being detected in parts of Wales. So it seems to be spreading, this disease, and we're sort of trying to understand why that is. And the symptoms that I mentioned earlier on all relate to sort of microbial symptoms, but there's a very high co-occurrence with the microbial symptoms of these signs of a beetle. So we can see in this first panel here, these D-shaped exit holes, which are sort of creatively named because they're very shaped like a D. In the middle panel here as well, if you kind of look in the bark a little bit more, we can see, if you look at this blue arrow here, the presence of these tunnels here, which are caused by insect larvae. And if you look at the red arrow, we've got the bacterial symptoms here. And this is just quite a good indicator that there's such a high co-occurrence of these insect symptoms alongside the bacterial symptoms. And the actual sort of insect that seems to be driving this is this 2-spotted oak-breasted beetle called Agrillus bigutatus. So here on the left-hand side, we've got the larvae of Agrillus bigutatus here. And on the right-hand side here is the Agrillus bigutatus adult, also sort of known as the jewel beetle because of its metallic colour. So the kind of key message I want to kind of get across today is that there's a really strong link between the bacteria that cause acute oak decline and these beetles. And we kind of basically want to figure out what this link is. And as I mentioned in the talk, I wanted to look at the odours. So the kind of research question that we're asking is, what is the role of leaf microbial odours, or volatile organic compounds, I'm going to call them from now on, in the beetle-bacteria interactions? So volatile organic compounds play quite a crucial role in ecological interactions. They're produced by a range of organisms, including plants, insects, and microorganisms. Some common examples that you might have heard of in terms of volatile organic compounds are, for example, insect pheromones. And yeah, these volatile organic compounds can play a role in communication between organisms. And the kind of reason why we're looking at them is because you can actually exploit these volatile organic compounds to sort of manipulate the behaviours of pests and pathogens. And what I kind of want to highlight to you is that these volatile organic compounds do already seem to be playing a role in the life cycle of the Agrillus bigutatus beetle. There's some previous work by Joseph Wutz, who's working at Rothamsted on this project as well. has shown that the Agrilis bigutatus use the smell of oak leaves to locate feeding and mating sites. And when the adults have mated, the females, which are now egg carrying, actually can smell bark as well. So they kind of move towards bark, they go down the tree, they move towards bark, and use these volatiles as sort of sites for egg laying. So this has already been established. These volatiles do seem to be playing a role in the life cycle of the beetle, but this is under a healthy tree system. And what currently isn't known is how the presence of this disease, acute oak decline, can influence the behaviour of these beetles and can be manipulated to improve the monitoring of the beetle. So the objectives of this talk, so I know the talk mentioned about bacterial odours, but we're going to talk a little bit about leaf odours at the start as well, but the focus mainly will be on bacterial odours. First thing we want to establish is we want to see if the presence of acute oak decline can cause changes basically to the smell of oak trees, and basically then want to see if the beetles can detect this change in smell. And in the second half of the talk, which will probably be the main part of the talk, is seeing if the bacteria which are produced by, the volatiles produced by the acute oak decline bacteria produce any volatiles, and do these have a role in the behaviour of the beetle. And it sounds like quite a niche topic we're looking at, but there is a sort of aim at the end of that, and that's to identify plant and bacterial volatiles which are attractive to the beetle, and this could be used to optimise semiochemical based lures, which can help with the monitoring of the beetle and can help with the management of acute oak decline. So yeah, as I say, the first part of the talk, we're going to focus on the leaf odours, and basically, does the presence of acute oak decline cause changes to the odour profile of the leaves? So this was some work that was done before I came back to Rothamsted, so I'm not going to take any credit for this, but it was a lot of work which involved a lot of field sampling around the United Kingdom. And what it basically involved was collecting volatiles from either healthy or declining trees from sites around the UK. So it's quite a sort of simple concept in that the leaves of these trees basically get wrapped in an oven bag. And with this silver kit here, which is an air entrainment kit, we can basically collect volatiles from the foliage over periods of time. So as I say, these volatile collections were performed on healthy trees and also trees that were diseased. And we did some analysis to see were there any differences in the compounds in a healthy tree compared to a diseased tree. And what was shown was that there was 4 compounds which seemed to be associated with symptomatic oak tree. So we've got these compounds A, B, C, and D. And with the dark grey bar here, which is the symptomatic oak, and the grey bar here, which is the non-symptomatic oak, these 4 compounds are significantly upregulated in the symptomatic oak, which indicates that they're potentially involved in stress. So from this sort of large body of work, we can conclude that there's 4 leaf volatiles, which are symptomatic signals of declining oak. And that is independent of where these volatiles were collected from the United Kingdom. The next thing we wanted to determine was whether these sort of stress-related compounds lead to any changes in the behaviour of the insects. So what we did was a technique called forearm olfactometry. And basically this is a choice test where the beetle can basically smell different treatments, and it will make a move towards any treatment that it likes the smell of. And we can record the amount of time it spends in each of these treatments. And from that, we can determine whether the beetle is indeed attracted to the smell of different stimuli. And in this experiment, we used our control, which was just blank air within this glass chamber, and our treatment, which is the oak foliage volatile. So we basically just took a set of leaves from a healthy tree in the first instance, just to show that the beetles are actually, they move towards the smell of the healthy oak foliage. As I say, these outputs are the amount of time that the beetle spent in each arm. And we tested virgin females and virgin males and found that virgin females and males both spent a significantly greater amount of time in the arm where they could smell the oak foliage compared to the blank air controls. So from this, we can establish that they kind of do seem to orient towards the smell of healthy oak foliage. As I said previously, this has already been shown, but we kind of just wanted to verify this before setting up the next experiment, where we're actually specifically looking at these compounds which are upregulated in declining oak systems. So we slightly modified the system. This time, one of the controls was healthy oak foliage on its own. And in the other treatment, we had the oak foliage volatiles, and we actually spiked the headspace above the oak with 3 of the 4 components that are associated with stress to see if this caused any changes in beetle behaviour. And interestingly, what we found was that the beetles spent a significantly greater amount of time in the arms which contained the smell of the oak foliage, plus these compounds which are associated with declining oak trees. So from the previous work, we've shown that the declining oak trees produce different volatile profiles to healthy oak. And with this experiment, we've actually shown that the beetles can detect the declining oak and they sort of orient towards it, which is quite interesting. So yeah, here we can conclude that virgin female beetles show significant preference towards the oak foliage plus the stress blend. So it could be that the beetles can kind of detect the declining trees and use that as a suitable site for egg laying. So as I said, the second half of the talk and the bulk of the talk is going to be on the bacterial odours. So think back to the introductory slide. I mentioned about the 3 bacteria which are sort of lead to the symptoms of acute oak decline. And we wanted to determine whether these bacterial volatiles influence insect behaviour and characterise the volatiles from these 3 bacterial species. So we did some more volatile collections here. So what we'll see on the left-hand side of your screens is the treatments that we had. So we have an uninoculated growth media as a control, and then the 3 bacterial species, Brunneria goodwinii, Rhinella victoriana, and Gibbsiella quercenicans, which are the disease-causing pathogens of oak decline. We then confine them in this glass vessel here and basically collect the volatiles from it. So this system basically pushes charcoal purified air over the top of the bacterial cultures. And you can see here we've got a glass tube. And within this glass tube, we've got an adsorbent polymer, which is called poropac. And air is then subsequently drawn out of the chamber, and all of the volatiles that the bacteria are making get stuck onto this polymer. And after a certain amount of time, we can then pass a solvent through this tube, remove the tube from the headspace, pass a solvent through, and we're left with this chemical extract, which basically contains the whole mixture of the compounds that the bacteria are producing. And the first thing we wanted to do is, before we do any sort of chemical analysis, is we want to determine whether these bacterial volatiles are actually influencing insect behaviour at all. So we use this technique, forearm olfactometry, where we basically, it's the same technique as I mentioned earlier, but it's a bit easier to demonstrate with these bacterial extracts. We have the beetle within this central chamber here, and then there's 4 glass arms which kind of come out of the arena itself. One of them with the treatment arm, so solvent extract that we collected from the bacteria goes into one of the arms, and then the other 3 are just left as solvent controls. And what we can do basically over time is measure the amount of time that the beetle spends in each of these arms, and we'll do it across several replicates, and we'll be left with information on how long the beetle has spent in each arm. And from that, we can determine, does it like the smell of these bacteria? And I've got a video here actually as well. Hopefully the technology works. Maybe Liz, you can give me a thumbs up if you start to see a beetle moving here. So we should hopefully be seeing, yeah, well, thumbs up. We should hopefully be seeing here the Gryllus bigutatus moving towards this treatment here, which contains the bacterial volatiles, showing that there is some interest in the smell of the bacteria. So yeah, as I mentioned, we had the 4 treatments, which was the control agar, the Brunneria goodwinii, Rhinella victoriana, and the Gibbsiella quercenicans. And if you look at the first graph in the first instance, which is just the headspace of the control agar, we can see that the beetles spent no significant difference in the amount of time spent in either the treatment or the control arm. So that basically means that the beetles aren't responding to the smell of the growth media on its own. But with the following 3 graphs, we can see that the beetles spend a significantly greater amount of time in the arm which contains the treatment, which is the different bacterial odours, compared to the control. From this, we can conclude there that the gravid female beetles show a significant preference towards these acute oak decline bacterial volatiles. And the next thing, we've done our behavioural tests, and our chemical ecology work is kind of driven by behaviour. So now we wanted to see what is driving this behaviour. And we use a technique called gas chromatography now to basically look at what compounds are in the headspace. So gas chromatography is essentially a technique which enables you to separate compounds out from complex mixtures, and we can see kind of what's in a sample. So these are 4 representative traces from each different treatment. So we've got the agar on its own, which is control, so no bacteria there. The Brunneria goodwinii, Gibbsiella quercenicans, and the Rhinella victoriana. What we're basically looking for is small kind of peaks in each of these treatments, and ideally ones that aren't present in the agar control, which would indicate that they're biological. And some of the key things that we were particularly interested in here is there's one compound that seems to be produced across all 3 bacterial species, so it seems to be a generic bacterial volatile. But interestingly, there seem to be 2 compounds here, which are called compounds 2 and 3, which only seem to be being produced by the Brunneria goodwinii. And what some studies have shown is that the Brunneria goodwinii seems to be one of the main sort of bacteria which causes the pathogenicity of acute oak decline. So the fact that it's producing these 2 specific compounds compared to the other 2 species is pretty interesting. So from this, we can conclude that there's one compound that's abundantly produced by all 3 AOD bacteria, and there's 2 compounds which are specific to Brunneria goodwinii. So all we now know is basically that the volatiles produced by acute oak decline bacteria seem to be attracting the beetle, and that the bacteria are producing volatiles. The next thing we need to do is basically link these 2 behavioural and chemistry techniques together to see which compounds are involved in the behaviour. And in order to do this, we use a technique called gas chromatography electroantenography, which is a bit of a mouthful, but I'm going to break it down so it's a bit easier to understand. What it is the antenna of the beetle is exposed to the volatiles produced by the bacteria. And if the antenna can detect any of these compounds, it will produce an electrical stimulus, which can be detected by the EAG recording. And you can link this with the compounds as they come out of the gas chromatograph, because it's kind of linked to gas chromatography. So hopefully this will make it a bit clearer. So we've got 2 example traces here, one for the Brunneria goodwinii, one for the Gibbsiella quaestionecans. And first, we'll just focus on the lower trace here. If you think about back to the previous slide, it looks pretty similar because this is the gas chromatography trace. So we can see we've got compound 1 here and then compounds 2 and 3. But the thing that differentiates this technique from the previous one is that we've also got this signal here, which is EAG. And we can see that we're kind of looking for where the antenna kind of dip, we see a dip in the signal. And we can see that for compounds 1, 2, and 3, there is this dip in the signal, which indicates that the antenna can detect these 3 compounds. As I say, compound 1 as well seems to also be detected by them. So from this, we can conclude that the volatiles produced by Gibbsiella quaestionecans and Brunneria goodwinii elicit antennal responses in the female Agrellus bigutatus. Thing is, that isn't the end of the story, because just because an insect's antenna can detect a compound, that's not to say that it's necessarily going to be attracted to it. It could be detecting a compound, but thinking, that doesn't smell very nice. I wouldn't want to move towards that. So the next thing we need to do is actually go back into our olfactometer, but actually purify out these 3 compounds and test them individually in this olfactometer to see if it is these compounds that are causing this behavioural change. So yeah, sorry, I jumped the gun a bit there. It's exactly the same as before, but rather than using the entire collection of volatiles by the bacteria, we're using these 3 compounds in their purified form, so we can actually figure out which compounds are causing this attraction. So the first one we tested was compound 1, which is the one that was produced across all 3 bacteria. And we can see that the beetle spent a significantly greater amount of time in the treatment arm compared to the control arm. We then purified out compounds 2 and 3. We tested these 2 together because it was technically quite difficult to separate them out individually because they kind of came off at quite a similar time in the trace. But we also tested them together because they are also produced together by the Brunneria. But we also found as well that the beetle spent a significantly greater amount of time in the arm which contains these 2 compounds compared to the control, which was just the solvent on its own. And finally, when we combine the 3 compounds together, we can see that the beetle spend a greater amount of time in the combination of the 3 compounds compared to when compound 1 is on its own. There's no significant difference between these 2 treatments, so we have to be a bit careful. But compared to the control, which is just the solvent on its own, the combination of the 3 compounds together seems to do best. So from that, we can conclude that the gravid female beetles show a significant preference towards these purified EAG-active bacterial volatiles. So in terms of the conclusions of this talk, first thing we've shown is that the Agrellus bigutatus can differentiate between healthy and declining oak foliage volatiles, and they seem to prefer the smell of these declining volatiles compared to healthy ones. Secondly, AOD bacteria produce volatile organic compounds, including these 2 Brunneria goodwinii specific volatiles. Gravid female beetles, when tested in the olfactometer, show a preference towards these AOD bacterial volatiles which also seem to elicit antennal responses by the beetle. And finally, that the gravid female beetle shows significant preference towards purified EAG active bacterial volatiles. So I've mentioned a lot about lab work in this talk, but I need to obviously talk about the applications of this work. In terms of what future work we might be planning on doing. We basically want to see can we actually see this effect under open field conditions. So we want to assess the effectiveness of bacterial volatiles in attracting Gryllus bigutatus through these semiochemical based lures in fields. So here we've got a semiochemical based lure, which we hope to optimize with the bacterial compounds we've shown to be attractive. Hopefully we can use it as a monitoring tool, so use the lures as tools to monitor the presence of the beetle populations effectively. So these can be deployed around the United Kingdom, particularly at sites where we're starting to see a northward shift of the beetle. And the sort of ideal aim is that it can be used as an early warning system for forestry managers. So it can provide them with an alert to the presence of the beetle, and this could sort of serve as a proxy for the presence of acute oak decline. So here we've just got an example picture of what one of these semi-chemical based lures might look like. And here we've got how one of these lures might be installed in a woodland. So I usually have to leave a bit of time at the end for the acknowledgements, because there's a lot of people to thank with this work, particularly our sort of collaborators at Rothamsted Research, so Joseph Butts and John Caulfield, Mike Burkett, David Withall, Godfrey Bangu and Pradip Songra, who are all involved in the work. Forest Research, particularly Sandra Dunham, Michael Crampton, Bridget Crampton, Megan Richardson and Nathan Brown. We've got some great collaborators at the University of Birmingham, James McDonald and Maureen Cambon, and the University of West of England as well, Carrie Brady. Just to mention as well, there's some great work that's been done in James and Maureen's work, where they've looked at the influence of the insect chemicals on the bacterial growth. So we've worked with them on that, which has shown some quite interesting results. And I'd also like to acknowledge the Backstop project, where a lot of this work was done. This was funded by the UKRI Strategic Priorities Fund and Programme on Bacterial Plant Diseases, funded by the BBSRC, DEFRA, National Environment Research Council and the Scottish Government. And then finally to thank Rothamsted, the Growing Health Institute Strategic Programme, and also forms part of the Smart Crop Protection Strategic Programme as well previously. So that's all from me, and happy to take any questions if anyone has any. Thanks for listening.
Thank you, Gareth. That's really interesting look at what's happening on the bacterial side, especially, and that all-important interaction in acute oak decline. We've had some comments coming in already and a few questions, but if you've thought of something, just, stick it in the chat or just put question in the chat and I will come to you. So the first person who's got a couple of questions and or one question and a comment is Anthony Mills. Anthony, if you're happy to come and ask your question, welcome you to ask it to Gareth in person.
My video doesn't appear to be working. Can you hear me?
We can hear you. Yeah. That's the important thing.
Sorry about the video. Oh, yes, there it is. Went in the wrong direction. Yes, the question was, looking at the map, there seemed to be a strip approximating to the limestone running from the south to or southwest of the northeast. And I wondered if there was any association with particular soils and the resulting drought stress of AOD.
Yeah, that's a great question. So I'm probably not the best place to answer that, but within this Backstop project, one of the work packages was looking at the influence of drought stress on acute oak decline. So there was a sort of an outdoor laboratory, basically, where different drought treatments were applied with the inoculation of these bacteria. I can't remember off the top of my head what the outcomes of that work package were, but I can sort of dig that out for you or point you in the direction of someone who might have a better answer. But there is definitely a link between, this combination of factors. I've spoken pretty much solely on the bacteria, but there's a whole combination of abiotic factors, like, say, soils and droughts, which can contribute to a tree's defences being weakened. But yeah, as I say, I think I'll try and find who you could speak to about that if you're interested.
Just your comment is fine. Thank you very much. No need to go further work. Thank you very much, Gareth.
Nice.
And then we had another question from Andy Evans.
Yep, that's great talk.
Gareth, I know it might be commercially sensitive, but are you able to tell us what the compounds are, or what kind of chemical group they might relate to?
Yeah, it's kind of commercially sensitive at the moment. We're planning to publish the work in the coming months. At which stage it will become publicly available. But yeah, we'll let you know when it comes out.
No, fine, no worries. Thought that might be it.
This one, I'll give people a little time for any more questions to come in if they think of anything. I was just really interested to hear how obviously the trees, the bacteria themselves can cause these lesions. And I think it's this interesting then, you know, the beetle's a native beetle, but suddenly being attracted to these trees. But I think I'm right in understanding that actually the beetle doesn't actually vector the bacteria. Is that correct?
That's something that's kind of undergoing at the moment. Is that some work has been done to try and figure that out, and I think there's some indication that the bacteria can survive in the guts of the beetle, but in terms of its role as a vector, I think that's still slightly unclear. Some of the work, I didn't speak about it today, but I mentioned about some of the work that Maureen and James have done at the University of Birmingham that we've worked alongside them with, showing that the beetle larvae can stimulate the growth of the bacteria and stimulate the virulence of the bacteria as well. Yeah, I was going to put a slide on that at the end, but yeah, I can maybe get that circulated because it's been released as a preprint. That work, so that's quite interesting. And there seems to be some specificity as well. When we looked at other insect larvae chemicals, they weren't triggering the growth of the Brinaria, but when it was a Grillus bigutatus, it was. So yeah, there's definitely a link. Yeah, as you say, that the vectoring is something that's kind of still underway, but this kind of other side of the work indicates that there could be other links that we're kind of still looking to manipulate.
I think, and I don't know whether you'll be able to answer this, Gareth, but it says a question from Richard Garnett. So if Richard is about, he's been.
Yeah, I'm here.
Hello.
Hi there. I'm very new to this, so apologies if my questions sound very naive. Are there any current treatment strategies apart from tree and disposing of it?
Yeah, that's kind of. We have actually attended a meeting last week where we kind of all came together and discussed what potential management strategies there are. That, you know, there's some things that are sort of thinning the canopy could be an option. I think one thing that people have been doing, I think, is felling trees, but that's not necessarily encouraged because even if a tree has the disease, it can still kind of provide a sort of ecosystem for other organisms. So yeah, that's kind of, I think, the next stage of the work. We've identified these management gaps, and it's sort of identifying what the research priorities are for that. So yeah.
Okay. I maybe offline, I'd appreciate a conversation. I've been treating a very old, about 750-year-old yew tree.
Mm-hmm.
Went through complete needle drop, and I've managed to get the needles to regrow on it.
Oh, right.
By applying a soil, an addition of a well-known fungicide to the root ball.
Oh, okay. Oh, that's interesting. Yeah. I'd be happy to talk more about that. Yeah, definitely.
Yeah. Okay. Well, maybe we'll get in touch outside the meeting.
Yeah, that'd be great. Yeah. Thanks, Richard.
Thank you.
So it's not a question.
Can I ask a question?
Yeah, go for it. Yeah, right.
So you might have mentioned this, Gareth, when you were at the start, but is AOD a new disease, or and if so, why is it a new disease?
I think it's relatively new in the grand scheme of things. I can't remember
evolutionary time.
Yeah. And I guess because it's such a complex range of factors that seem to be involved in it, that seems to be kind of why it's quite unknown. What we're sort of seeing in the literature at the moment is there's more reports of it across Europe. I think recently somewhere in Slovakia had reported it. And I suppose because it's such a complex disease and it's not just one bacterium necessarily that's causing it, it's very hard to kind of characterise and diagnose. Sandra's team at Forest Research have done a lot of the main work in characterising the disease, particularly in the United Kingdom. So I'd say it's a relatively new disease, but still kind of, I think, I can't remember when their work started. I wouldn't want to hazard a guess either, but yeah, relatively new, I'd say.
And just a note, I put a link to the preprint you mentioned in the chat there. So if anyone wants to download that that work, which has just been released a couple of weeks ago, or a month ago. So.
Thanks very much.
So just to kind of, oh yeah, Gareth has just put something in the chat, but so the acute oak decline as this beetle bacteria interaction was distinguished from other oak declines in early 2010. So it's only about sort of 10-15 years that we've really recognised this specific complex in oak declines. There are obviously other situations in which oak trees decline, but recognising this bacteria beetle kind of combination is actually relatively recent. So yeah, just to kind of clarify that. And there's a couple of people also, Louise and Gareth in the chat, have said about best not to fell the trees just because they have acute oak decline unless they're posing obviously a health and safety risk, you know, if they're over a public footpath or something and they're likely to fell branches. Some trees can go sort of into remission and recover from the tree. So just from a sort of management aspect, that's something to bear in mind. I know there's been a lot of work done on the management in terms of ash dieback, for example, the same sort of thing, not always felling trees just because they have the disease. Louise has just posted in the chat the Forest Research information link on that. So thank you, Louise. It doesn't look like there are any other questions coming in. It's been absolutely fascinating to hear about this work. I think it's not very often we kind of hear things on volatile compounds, and definitely not on bacteria. It's normally fungi in the
I think Anthony has got a finger up he'd like to ask.
Okay, sorry.
Sorry, there's no raised hand, I don't think. A couple of things. I was on a citizen science survey at Cambon and Ickworth with Nathan Brown, and I believe he said that approximately 30% of oaks can survive AOD. So definitely, unless they're obviously out, then don't fell in first instance. Secondly, the article posted from a very quick skim of the abstract seems to indicate that the larvae stimulate the production of these bacteria. So I'm just, what is the evolutionary advantage to them of having these bacteria in the tree? How does it assist the beetle?
That's a good question. And what I think has been kind of recently shown is that it could relate to the sugar content of a diseased tree. So there's sort of early indication that diseased trees might have higher sugar content. So if the beetle is attracted to these declining volatiles produced by these declining trees, it could be that there's a nutritional benefit for the beetle. But actually, what would be interesting to look at is the actual nutritional content of an actual lesion as well. Because if we're hypothesizing that the bacteria within the lesion are producing volatiles that the beetle then move towards, it could indicate that these lesions themselves are rich in sugars and amino acids that might be beneficial to the beetles. That's a really good question, yeah.
Wow. Thank you very much for that. Thank you.
All right, no worries.
Hey, I think that's all the questions now wrapped up. Thank you so much to Gareth for giving a talk today. As Gareth said earlier, this is the second talk we've had from the Applied Forest and Tree group with the AAB. We're always looking for people who might like to give a talk to this group. So if there is something you think would be of interest to our membership, then please do get in touch. Is there anything else from you, Gareth?
No, just to say this has been recorded, so after I chat with Gareth about it, then if he's happy, then we'll load it onto YouTube and do that. But I'm not guaranteeing that right now because we haven't talked about it. But if so, then it might be available for you to pass on to colleagues if they're interested in it as well. And I think, as Elizabeth said, we'll hope to have another webinar sometime in the new year.
Could you? It doesn't appear to be able to can't copy the chat. Could you please include a copy of the chat if you post it online?
Yes, indeed. Yes.
Thank you. Thanks very much.
Thank you again, Gareth.
Thanks very much. Thanks for listening,
Thanks,
everyone.
Gareth.